Hello, my name is Mallory Brayer and I'm from George Washington University. I'm working on a project to characterize mechanosensitive channels that may be responsible for ultrasound-induced insulin release in pancreatic beta cells. Beta cells are part of glucose homeostasis. In general, they respond to increases in blood glucose. So for instance, after a meal when there's excess glucose, these cells can use a specialized transport protein known as GLUT2 to bring in this glucose. Once inside the cell, it can be metabolized into ATP that can then inhibit potassium ATP channels. This blocks the inward flow of potassium ions into the cell. The blockade of this causes depolarization within the membrane of the cell and activates voltage-gated calcium channels. Activation of these channels allows calcium to flow into the cell. Once inside the cell, this calcium can interact with stored insulin granules and promote their exocytosis. Once outside the cell, this insulin can then go to peripheral tissues like adipose or muscle tissue and promote these cells to actually take up this excess blood glucose and either use it for energy or store it for future use. Many people know beta cells, insulin, or even sometimes glucose in the terms of diabetes. Specifically, one of the most common forms, type 2 diabetes. This is characterized by chronic hyperglycemia. Although there are multiple causes that go into the development of this disease, including things like peripheral resistance to insulin, there's also usually some disruption in this insulin secretion process that leads to this chronic hyperglycemia. One of the most common ways to treat this disease is through lifestyle changes and diet changes. However, this doesn't work well for everyone, and some people continue to still see this hyperglycemia. So many people rely on pharmaceutical interventions to treat it. Many of these medications do carry side effects ranging from weight gain to cardiovascular events. So one of the main goals of our lab is to try to find an additional treatment that may have less side effects. Specifically, we are working with unfocused ultrasound to see if we could treat type 2 diabetes with it. Preliminary data from the lab has shown that therapeutic unfocused ultrasound can induce insulin secretion. And this work was able to show that there are certain parameters that do promote insulin release without harming cells. As we can see, this 1 megahertz and 800 kilohertz frequencies aren't causing a significant decrease in cell viability, meaning the ultrasound is not harming the cells. At the same time, though, the 800 kilohertz frequency is able to cause this release of insulin into the extracellular environment. The work shown here was done in an INS cell model, which is a rat insulinoma cell line and a common model used to study diabetes. This work has further been replicated in ex vivo rat pancreas as well as in vivo in mice. But we really haven't done much with human samples yet, so we're not sure if this exact same mechanism would occur, and how we also don't know the mechanism that's causing it in general. Studies focused on islet swelling have identified a few mechanosensitive channels that may be present in islets, specifically piezo-1 and TRPV2. Both of these studies have been done in rodent models and were not focused on ultrasound, but instead focused on cell swelling. The aim of this project is to identify mechanosensitive channels that may be expressed within human pancreatic islets in human samples, as well as working to further characterize any that may be expressed in these INF cell lines. We hope that by understanding what channels are actually there, we might be able to uncover the mechanism that drives this ultrasound-induced insulin secretion and have a better understanding of it. In order to do this, we were using two types of cells. We had primary tissue in the form of human islets, as well as our cell culture, the INS insulinoma cell line. There were three treatments that were used on the cells, the first being our control. In this case, cells were kept at a low glucose concentration because that should not have any stimulatory effect on them. We also had a high glucose treatment because these cells in their physiological environment respond to high glucose, so this may have some effect on them. And our ultrasound treatment was done for 5 minutes continuously with unfocused ultrasound at 800 kilohertz frequency and a 1 watt per centimeter squared intensity, basically just using these parameters that had previously been shown to not harm the cells and to promote insulin release. All ultrasound was carried out within a water bath to minimize the thermal effects that we would see from 5 minutes of continuous ultrasound. Once cells were treated, we then isolated RNA from them so we could start seeing what genes may actually be expressed by these cells. So we specifically did reverse transcriptase and quantitative PCR in order to see what mRNA was available within these cells and if any of these mechanosensitive channels of the piezo or TRPV family were being expressed. Once we found genes that were being expressed and found mRNA of them, we were able to then do more targeted studies looking at just those specifically expressed genes and work at a protein level. We isolated total intracellular protein from treated cells and run these on a Western blot in order to see if this mRNA that was being expressed for these mechanosensitive channels was then translated into actual protein that could be used by the cell. There were a few additional studies done with confocal microscopy within the INS cells just to see the potential localization of these mechanosensitive channels. Our main finding was that we were able to find several different forms of mechanosensitive channels within human pancreatic islets. Specifically, we were able to find TRPV2 and piezo1 similar to the previous rodent models, though these forms seem to be pretty widely expressed across different animals then. We were also able to find a TRPV5 isoform of the TRPV family that was expressed within human pancreatic islets. We also saw that regardless of whether we had these cells in a low glucose or high glucose and stimulatory environment, we did not see a significant change in the amount of mRNA or gene expression of these specific channels. We then investigated these channels further at a protein level and ran Western blots to see if we could still detect these proteins, if they were transcribed, as well as if their expression had changed due to different treatments. What we saw was that we were able to identify TRPV2 and TRPV5 at a protein level, and that neither high glucose, so our physiological stimulus, or ultrasound, our potential therapeutic stimulus, caused a change in the amount of these mechanosensitive channels. We further did some confocal imaging of these INS cells, a common model diabetes study, to see if we could find any localization of these channels. Given that they are channel proteins, we would expect them to be within the plasma membrane. And the plasma membrane was stained in green here, so that we were able to see if mechanosensitive channels, in this case piezo1 stained red, had any localization around this area. And in both cases for piezo1 and TRPV5 within the INS cells, we were able to see some degree of localization towards the plasma membrane, suggesting that they are expressed there and supporting their role that they are interacting with the extracellular environment. The main conclusions of this presentation were that we were able to identify TRPV2, TRPV5, and piezo1 as the mechanosensitive channels that are expressed within human pancreatic islets. We were also able to demonstrate that neither our potential therapeutic treatment of ultrasound nor physiological conditions of high glucose would alter their expression. We were also able to show that there was localization of TRPV5 and piezo1 towards the membrane of these INS rat insulinoma cell lines. To continue this project, we hope to do some confocal imaging of islets to see if we see a similar pattern of localization of these mechanosensitive channels within them, as well as do some functional studies to investigate the roles of mechanosensitive channels as a whole, as well as targeting specific isoforms of these channels to see what their specific role within this process is. Thank you.